Antonio Quadrato from Autonomous University of Madrid. I want to thank Antonio for his continuous support to the webinar series, and I also want to give thanks to the all organizing committee high-level webinar series, in particular to Professor Luciano Sasso from Sapienza University of Rome, that spearheaded the project of the webinar series in the antioxidant.it platform. Before introducing our highly esteemed speaker, who will be joining us in today's webinar with their experience and knowledge in oxidative stress and aging field, I want to introduce Professor Roberta Foresti, member of our cross-section, and I'm honored to share the chair with a so brilliant scientist. Roberta is a professor of biochemistry at the University of Paris-Sacré-Cœur and working as a scientist at the Mondor Institute of Biomedical Research in France, focusing on research on oxidative stress, energetic metabolism, and drug discovery. Briefly, I want to remember that each of us speakers will give a talk of about 20-30 minutes. The webinar will last up 2 hours. The discussion will be open and after the 3 talks. Please feel free to ask a question in the chat at any time, not be shy. When doing so, please don't forget to mention your name and affiliation and to whom the question is addressed. The webinar will additionally streamed online upon registration. Now it's my pleasure and honor to give the floor to Professor Roberta Foresti. And please, Roberta, introduce our speakers.
Hello to everybody. Thank you so much, Brigitta, for inviting me today to share this session with you. And I need also to thank Antonio, who kindly suggested my name, since he's not able to join us today. And I hope to step into his shoes and, to make him proud of this. So it is, I think it's going to be a very interesting afternoon. And I'm very interested on the topic of aging. And the first speaker that we are going to have is Anna Groho from the Jagiellonian University from Krakow in Poland. And the title of her presentation is NRF2 and Aging of the Vascular System. The floor is yours, Anna.
Thank you, Roberta, very much for this kind introduction. I also want to thank Brigitta for her kind invitation. And now I will try to share my screen with you, and please just let me know. Whether we can see it in a full screen mode. Right?
Good.
Okay. Yeah. Thank you. Okay. So today I would like to talk about NRF2 and aging of the vascular system, and I would like to share with you our data in this subject. And I think there is no need to introduce NRF2, but just to stay on the safe side, I will briefly mention that the NRF2 is a transcription factor that is repressed by KEAP1 protein and in the unstressed conditions, the NRF2 undergoes constant degradation through proteasome. However, in the presence of oxidative stress or in the presence of electrophilic compounds, it is not degraded, and de novo translated NRF2 translocates into the nuclei, where upon binding with its consensus sequence, the antioxidant response element, it transactivates the expression of many cytoprotective genes. And I believe that we all work with the oxidative stress, most of us probably with NRF2. And today I would like to share with you our data concerning the role of NRF2 and KEAP1 in the cardiovascular system, in the cardiovascular diseases in relation to aging. And as you know, cardiovascular diseases are very strongly associated with aging, and there are many risk factors for the CVDs. However, the aging is the non-modifiable factor. So actually there is nothing we can do about the aging itself, but maybe we can modulate some pathways to to prevent the cardiovascular disease development. And there are at least several proteins which are very strongly associated with aging. These are mTOR, FOXO3A, PARP1, SIRT1, and also NRF2. And this relation of NRF2 and or the association of NRF2 with aging can be seen, for example, in this guy. This is the naked mole rat. So the lifespan of this rodent is more than 30 years, and the level of NRF2 is around 6-fold times higher than, for example, in wild-derived animal. And also for the worm and also for the fruit fly, there is a strong association between the lifespan and the level of the NRF2. And it can be also visible when we compare the lifespan, the maximum lifespan of those different rodents, and in terms of the NRF2 activity, and you can see there is clear correlation between the NRF2 activity and the maximum lifespan of the rodents, which is not the case, for example, when we compare the NRF2 activity with the body weight. So it seems that there are some associations between the NRF2 and aging in different species. So our goal was to address the question of NRF2 and the physiological and premature senescence mechanism that can be seen in the vascular system, and in particular in the murine aorta and also in human aortic endothelial cells. And what we found was that the level of the NRF2 protein is strongly decreased in the endothelial cells isolated from the aged donors. These are the human donors of the endothelial cells. So these endothelial cells are isolated from the aorta. And we saw that really strong decrease of the NRF2 protein level. On the other hand, when we took the endothelial cells isolated from the young donor, we observed that upon knockdown of NRF2 in those cells with siRNA, these cells underwent premature senescence. And also we observed the senescent phenotype in the NRF2 knockout animals, the young animals. They were around 2 months old. However, in the knockout animals, we observed that those aortas are prematurely senescent. So we were wondering why NRF2 protein level is decreased in human physiologically aging endothelial cells. What is the mechanism of premature senescence of NRF2 deficient cells? And also we were wondering how NRF2 deficiency may affect the age-related cardiovascular diseases development. And in this case, we were mostly focused on the abdominal aortic aneurysm. But addressing the first question, which is why NRF2 protein level decreases with age in human endothelial cells, we did a very simple experiment comparing at first the protein level, so it looked like that, but then we had a look on the transcript level of NRF2 to see whether this deregulation can be seen also on a transcriptional let's say, regulation. And as you may see here, actually there is no difference between the transcript level of NRF2 between the young-derived endothelial cells and the aged donor-derived endothelial cells. So it seems that this regulation is rather post-transcriptional. And we found that actually the level of the protein can be regulated by microRNAs. And there are several microRNAs that are related to aging, and one of them is microRNA 34A. And we observed that the level of microRNA 34A is significantly increased in endothelial cells isolated from aged donors. And this is associated by the upregulation of P53, which is a transcription factor directly transcribing microRNA 34A. So probably that could be the reason for this decreased, let's say, level of NRF2 protein in aging endothelial cells. To address this point, we treated the cells with p53 alpha, which is the inhibitor of p53, and at the level of the transcription, we could observe that the transcript of NRF2 was upregulated both in young donor-derived cells and also in aged donor-derived cells. After 3 hours, the effect was lost after 6 hours. We have no data at this moment regarding the level of the protein. However, and this is the really fresh ongoing story. However, what we can say right now is that the low level of NRF2 protein in physiologically aging endothelial cells is related to the post-transcriptional mechanism, and we hypothesize that the role for the microRNA 34A, and we are going to check it soon. So I believe that I will be able to share this data with you soon. Regarding the second question that we asked about the mechanism of the premature senescence that is induced by the NRF2 deficiency, at first we had so this, just to remind you, so this effect can be seen both in the human cells and also in the murine aorta. And at first we had a look on the oxidative stress that we supposed could be the trigger for the senescent phenotype, because we know that oxidative stress can induce the premature senescence. However, looking at the level of the expression of the antioxidant genes between the control cells and NRF2 knockdown cells, we couldn't observe much. Sorry? We couldn't observe much difference for the GPX1. There was some slight decrease in the level of SOT2 and catalase. We saw the trend to increase the level of the superoxide anion. However, this was not the significant effect. And when we compared the level of the lipid peroxidation between the control cells and the NRF2 deficient cells, we couldn't observe any difference, whereas the cells exposed to iron as a positive control showed a very potent lipid peroxidation. Moreover, we were not able to reverse the senescent phenotype by the incubation of cells with temple compound, which is the scavenger of the superoxide anion. So to our surprise, actually, we didn't see any oxidative damage in NRF2 deficient cells, which was quite surprising effect. So looking for the reason for the senescent phenotype of those NRF2 deficient endothelial cells, we had a look on the nitrosative stress, because apart from the oxidative stress, also the nitrosative stress can trigger senescent phenotype. And we were mostly interested in protein S nitrosation, since we observed some deregulation of nitric oxide, let's say, compartmentalization between the NRF2 deficient cells and the control cells. And the S nitrosation is a post-translational modification of the proteins, which is the covalent attachment of the nitrosa group to cysteines, to thiols. And this is the reversible process that can be reversed by 2 enzymes, thioredoxin reductase and GSNOR. And we compared the level of S nitrosation between our control cells and NRF2 deficient cells, and we observed a profound effect that was visible in NRF2 deficient cells. And actually, we also could detect very strong protein S nitrosation in the murine aorta in NRF2 knockout animals. So asking the question about the functional relevance of S nitrosation, we treated the cells with DTT, which is the specific reducer for S nitrosated proteins. And you may see here that this treatment was successful. There were no S nitrosated proteins observed in those lysates. And this treatment led to the very strong lipid peroxidation, but only in NRF2 deficient cells, which was associated with the cell apoptosis and cell death. So this showed that this S nitrosation could protect NRF2 deficient cells from oxidative damage and also from apoptosis. So we wanted to know which of the proteins that undergo S nitrosation could be responsible for that. So we did the mass spec analysis of S nitrosated proteins between the NRF2 deficient and control cells. And we identified one protein that was related to redox biology, and that was NOX4 and ADPH oxidase 4. So in the next step, we checked whether this is the protein that undergoes S-nitrosation that could be responsible for the protection of NRF2 deficient cells from oxidative damage and from apoptosis. And we knocked down NOX4 in NRF2 deficient cells, and we treated those cells with DTT. So you may appreciate that indeed NOX4 and S-nitrosation of NOX4 could protect NRF2 deficient cells from oxidative damage and from apoptosis. So that was the conclusion from this part of our studies, that the S-nitrosation of NOX4 protects NRF2 deficient endothelial cells from oxidative damage. And our next question was how S-nitrosation was created, because we know that in the condition of, let's say, the cellular environment, it is quite difficult to create the NO+ cation. And previously, the S-nitrosation was thought to occur on the non-enzymatic pathway, but taking into account those concerns regarding the cellular microenvironment, there were some doubts, let's say, about the creation of or generation of S-nitrosation on the proteins. So we observed that the NRF2 deficient cells have the increased level of KEAP1, so the repressor of NRF2. So we have hypothesized that the KEAP1 could be that is overexpressed here or overabundant when NRF2 is lost, could be responsible for the S-nitrosation of those proteins. So we knocked down KEAP1 in NRF2 deficient cells, and we confirmed with 3 different methods. Here you can see the immunofluorescence, also the Western blotting, and also the most specific one, which is the hemiluminescence, that S-nitrosation in NRF2 deficient cells is mediated by KEAP1, because deletion of KEAP1 from those cells completely reverses S-nitrosation of these proteins. Moreover, when we did the KEAP1 pull down, we identified at least 2 transnitrosating proteins, so the proteins that can transmove the, let's say, NO from one protein to another. Among them, GAPDH is such a protein. Another one is protein disulfide isomerase. So those 2 pull down together with KEAP1. And in case of the protein disulfide isomerase, we couldn't confirm that that could be we couldn't confirm this effect in further experiments. However, what we observed for the GAPDH was that GAPDH co-immunoprecipitated with KEAP1 and together with nitric oxide synthase. So this showed that the sort of the complex is formed in NRF2 deficient endothelial cells. And knocking down of any or actually knockdown of each of the proteins that are complexing together. So in this case, we tried nitric oxide synthase 2, nitric oxide synthase 3, GAPDH or KEAP1. Knocking down each of these proteins decreased the level of S-nitrosation in NRF2 deficient cells. So those pictures here, when we compare them to the level of S-nitrosation seen here in NRF2 deficient proteins, here this S-nitrosation is much lower, which gives us the let's say the answer that this complex could be responsible for the overabundant S-nitrosation seen in NRF2 deficient cells. And apart from that, we observed that knocking down KEAP1 in NRF2 deficient cells brought back the endothelial cell proliferation. We observed downregulation of P21, and also we reversed the effect of the senescence phenotype. So in summary, we found that the NRF2 deficient endothelial cells are senescent due to the overexpression of the unrestrained KEAP1 that is not kept inbound, let's say, by NRF2 in the cytoplasm, and this effect is associated with a potent S-nitrosation of the proteins. And finally, we wanted to know what is or what could be the effect of NRF2 deficiency on the development of the abdominal aortic aneurysm. So we were working with the aneurysm in our also previous studies. And as you know, it is really complicated and could be fatal vascular disease. This is the permanent dilation of the aorta, and quite often it may happen that it leads to the aortic wall rupture, and around 80% of such cases may unfortunately result in inpatient death. So there are at least several factors, among them age, male hypertension, smoking are the most important, I believe. So what we found was that the correlation between the level of NRF2 target genes and the AAA diameter in human samples. So we observed that the level of, in this case, GCLM and NQ1 expression gets lower as the AAA diameter is getting higher. So there is this correlation in human samples. And then we induced the animals, wild type and NRF2 knockout animals with angiotensin II to induce AAA formation. And we observed that the lack of NRF2 signaling favors AAA formation in mice. You can see it here. So this is the number of incidents between wild type and knockout animals, and you may also appreciate this picture of the aneurysm detected with the ultrasound. And what we found was that upon injection of angiotensin to animals, we observed upregulation of the microRNA 34A exclusively in the endothelial layer of the aorta. So you can see it here with the in situ hybridization. And we also found that the microRNA 34A level is increased in the sera of mice treated with angiotensin II. However, the highest levels could be found in those animals that had developed AAA, comparing to those who had not. So this gave us the sort of the hint that actually microRNA 34A maybe could play a role in a AAA formation. So at first we wanted to have a look on the crosstalk between the micro 34A, NRF2 and aging. So we compared the level of micro 34A in endothelial cells derived from the aged patients and the young patients, and we observed that there is a strong increase in the level of micro 34A, and also p53 was upregulated. The introduction of micro 34A to young donor-derived endothelial cells, that you can see that we confirmed the overexpression of microRNA 34A, led to the induction of the premature senescence of those cells that was associated with the increased levels of several pro-inflammatory proteins that are or could mediate this senescent secretory effect. And then we compared the level of microRNA 34A in control cells and NRF2 deficient cells. And again, we confirmed that the level of this microRNA is increased, and this effect was mediated by p53. And this microRNA was responsible for the induction of the senescent phenotype of NRF2 deficient cells, since the knockdown of micro 34A completely reverse the effect on the premature senescence. And then we want to have a look how it looks in NRF2 knockout animals. Do you remember that they also show the senescent phenotype in the aorta? And we did the in situ hybridization. So this is the on-fast projection of the murine aorta. And you may appreciate that the level of micro-34A is increased in endothelial cells in the lumen of the murine aorta. So to address the question whether micro-34A can be responsible for the increased number of incidents of AAA in NRF2 knockout animals, we breed those three genotypes of the animals, so the NRF2 knockout animals together with the microRNA floxed animals and together with mice expressing Cre recombinase, which is regulated by the promoter V Catherine promoter, so the promoter specific for endothelial cells. So upon many crossing of these animals, we ended up with this animal, so the NRF2 knockout animal with the deletion of microRNA specifically in endothelial cells. And at first, we confirmed that we have the knockout of microRNA 34A in the endothelial layer of the aorta. So here are these are the endothelial cells. Here are the smooth muscle cells. So you may see that this model works nicely. And this deletion of micro-34A endothelial layer protected the endothelial cells from the senescent phenotype. You may appreciate here that the more white color means the, let's say, the healthy endothelium, whereas the dark color means or the black color means the premature senescence of the cells. And such animals with the deletion of micro-34A in endothelial layer, NRF2 knockout animals, these animals were completely protected from AAA development. And also, so these were the young animals. And here we tested the 6-month-old animals. They were wild type in terms of the NRF2. However, deletion of micro-34A in the endothelial layer protected the cells from AAA development. And this effect was not associated with, there were no differences, for example, in a blood pressure level between those two genotypes. It was not associated with some, let's say, troubles or problems with the signaling coming from the angiotensin II receptor, and it couldn't be attributed, for example, to the some deregulation of the angiotensin 1,7 metabolite, which has the opposite effect comparing to angiotensin II. So this effect couldn't be attributed to some problems with, for example, with the transduction of the signal coming from angiotensin II. So just to cut the long story short, we also had a look on endothelial function in those animals with the deletion of micro-34A in endothelial layer, because we thought that maybe in this case the animals are protected because the function of endothelium is improved. However, we could observe deterioration of the endothelial function in these animals. So also this protection in terms of the AAA formation couldn't be attributed to the endothelial function. However, what we found, and that was quite interesting for us, is that in those in this genotype, with the deletion of micro-34A in endothelial cells, we found that in the mice injected with angiotensin II, we observed like a quite strong proliferation of endothelial cells. found in the aorta. So we thought that maybe endothelial cell proliferation could be responsible for the protection of those animals from AAA formation. So to address this point, we treated the cells with rapamycin, which is the which is of course the inducer of autophagy, but apart from that, it was known that it can inhibit endothelial cell proliferation. And indeed, in this case, the animals treated with rapamycin comparing to animals treated with the vehicle showed that the staining shows that endothelial cell proliferation in the aorta is inhibited with rapamycin, what you can also see here in this graph. And the animals treated with rapamycin were, in this case, this effect, protective effect of deletion of micro-34A in endothelial cells was reversed by rapamycin treatment. So those animals were very, developed a lot of aneurysm, and those aneurysms were quite prone for rupture. So it seems that the endothelial microRNA 34A may promote AAA formation, and it may promote this formation both in NRF2 knockout animals and also in wild-type animals. And the rapamycin inhibits endothelial cell proliferation and aggravates AAA formation. And to we were trying also to look for the mechanism that could be observed behind. And to address this point, we did the mass spec analysis comparing the proteome of aged donor-derived and young donor-derived endothelial cells. And we observed that a lot of proteins related to proliferation were downregulated. And when we did the knockdown of microRNA 34A in the aged cells, we observed that those proteins could be upregulated, and actually most of the proteins that were significantly changed here in those analyses were related to cell proliferation. So we wanted to confirm whether this is something that can be also seen when analyzing the proliferation itself in those cells. So we observed that the knockdown of microRNA 34A in cells led to the increase in the proliferation. And also we observed that the stimulation of endothelial cells with angiotensin II, like we observe in the animal model, led to the increase in a number of cells that were proliferating. So that shows that actually the same effect could be observed for the in vivo model and also for the in vitro model. And among those proteins that were deregulated here that could be that could mediate the effect of microRNA 34A, we identified MTA2 as an interesting protein. So it is decreased in the aged endothelial cells, and its expression is brought back upon deletion of microRNA 34A. And we wanted to validate it, whether this MTA2 protein, which is normally responsible for the deacetylation of histones, it's a direct target of p53 signaling, whether it can be the mediator of the effects that we see in microRNA 34A deficient cells. So we confirm that its level is increased in response to angiotensin II treatment, both in control cells and also in cells that are devoid of microRNA 34A. So you may appreciate here in this graph. And this experiment that is presented here shows us that the treatment of cells with angiotensin II increases the proliferation rate of endothelial cells, and the only group of cells in case of which we observed the inhibition of proliferation. These were the cells devoid of MTA2 and also the microRNA 34A. So this shows that MTA2 can be the mediator or is a target of microRNA 34A that is responsible for the regulation of endothelial cells proliferation in response to angiotensin II. So summarizing, we can say that the low level of NRF2 protein that we can see in a physiologically aging endothelial cells is related to the post-transcriptional mechanism, and we hypothesize that this effect could be dependent on micro-34A, and we are going to check it. NRF2 deficient endothelial cells are senescent due to the overexpression of the unrestrained KEAP1. And this effect is associated with a potent S-nitrosation of the proteins. We also found that the S-nitrosation of NOX4 is responsible for the protection of NRF2 deficient endothelial cells from oxidative damage. And we also found that the endothelial micro-34A promotes AAA formation in NRF2 knockout mice and also in wild-type animals, aging wild-type animals. And we suppose that this mechanism can be related to the regulation of endothelial cell proliferation, and it can be related to the MTA2 as a target for the microRNA 34A. And finishing, I would like to acknowledge the people who were involved in the study, and I will be happy to take any questions. Thank you so much.
Thank you very much, Anna. So I think you gave us an excellent lecture, and we will wait for the end of the session for all the questions. So we will move on to the next speaker, Professor Paul Shields from University of Glasgow in the UK. He's going to present us a seminar entitled Manipulating the Exposome of Aging from the Environment to Fundamental Cellular Mechanisms. Thank you, Paul.
Okay, thank you. Can you hear me?
Yes.
Thank you for the invite. I'm delighted to speak. This is a slightly different topic, but very synergistic with what Anna has been speaking of. So today I will tell you about work on my lab on the exposome of aging. I will tell you about how to measure aging and the rate at which you are aging, and then manipulating the environment that you live in to slow down the rate at which you age. So in honor of Yanis, I will start with something Greek, Hippocrates, who once said, "Let food be thy medicine." That's actually a misquote. There are two things in science, he said. Two things, actually, science and opinion. The former begets knowledge, the latter ignorance. And we really do need knowledge about how we age, because currently it's a topic of great interest because we realize that aging is malleable. We can slow it, but often the interventions required to slow it down are oversold, and they do not have a good evidence base. Why aging is of great interest presently is because the demographic of the aged in the world, so those over the age of 60, is now greater than at any time in human history, and that demographic is growing. Unfortunately, despite the fact that we can keep more people alive for longer, we cannot keep them alive in good health, and the gap between our years of healthy living and our actual lifespan has not been reduced. This has attracted a lot of attention from governments and pharmas because the cost of treating non-communicable diseases has been estimated pre-COVID to be around $47 trillion between 2010 and 2030. And that amount is expected to be much larger now post-COVID. Again, the economic impact, for example, within Europe of treating non-communicable diseases and dealing with social inequalities in health is staggering. It's around 9.5% of GDP. This is an issue that will only grow in developing parts of the world because the effects are amplified by social inequalities in health linked to social deprivation. So if we look at where the population of the old is growing in the planet, it's generally in the global south, there we will see a tsunami of diseases of aging that we already observe with prevalence in more affluent areas of the world, in particular Europe, the US, and Japan. What typically we are expecting is that we will see the emergence of this diseasome of aging. They're all very different types of diseases, but they all share a common underpinning component of a dysregulated aging process. That diseasome has a number of common features, which are also of note affected by a changing environment. And this includes diminished stress defenses, principally mediated and controlled by NRF2, loss of diversity of gut microbes, a greater inflammatory burden, and a loss of physiological resilience. Typically, the diseases within the diseasome of aging are treated as separate disease modalities, and we get a small improvement in our health span. So essentially, we live 10 to 15 years at the end of our life with a range of different morbidities, and we are managing our death. If, however, we target the underlying components of the aging process, then we can preempt the onset of these diseases of aging. And this is the geroscience hypothesis. So it's tackling the tractable components of aging to compress the period of morbidity at the end of your life, and therefore increase your health span or years of healthy living. This is of relevance to me in the UK and the city of Glasgow, because if you look at deaths per 100,000 in Europe, this is Scotland, the sick man of Europe, and Glasgow, of course, is the sick man of Scotland. Scotland does not do very well in comparison to the rest of the continent. However, if you look by council areas or prefectures within the city and across Scotland, we can see that the picture is skewed by these three areas. And this is the east end of the city of Glasgow, where there is a very steep socioeconomic gradient and a greater prevalence of diseases of aging. And we're trying to understand why, and can we actually intervene appropriately. So we've been trying to model this with colleagues over the past few years, to take people who are on a poor trajectory of aging here, shown in red, and as they get older, bring them up to one that is better, illustrated in black, where the individuals are physically and physiologically more capable and robust and less frail. So that means understanding an interplay between our genetics and epigenetics and our environment, always bearing in mind that we have to view this under the umbrella of antagonistic pleiotropy. So that is, what is good for you when you're young is not necessarily good for you when you're old, and vice versa. So knowing when to intervene to change this is important. So aging is highly individual. And I always like thinking of it in terms of the opening lines of Anna Karenina. All happy families are alike. Each unhappy family is unhappy in its own way. We age unhappily. All of us are aging at different rates, accumulating physical and physiological deficits in different ways, with a variety of rates for different organ systems. So we are accumulating wear and tear in a bespoke fashion. The aging is a process that starts pre-conception. It starts when your mom is in your grandmother's womb, and part of you is an egg cell in your mom. So the lifestyle and exposures of your grandmother will impact you and your mother. And your mom, when she is pregnant with you, her lifestyle and exposures will also affect you. So you can have effects that jump generations. And this is often difficult to either appreciate or anticipate or investigate. However, what we do know is that aging is not passive. It is actively regulated by genetic pathways. and understanding the molecular basis of these is necessary for their manipulation to give us more years of good health. And this, of course, is the Shields trajectory of aging, quite unique and a bit disturbing when I see myself over time, but touch wood, I still remain physically capable, but I'm anticipating morbidity in the future, unfortunately. At present, we can hallmark aging into features that are common across taxa, so common to flies, to worms, to yeast, to mice, and to humans. And these are features such as telomere attrition, cellular senescence, mitochondrial dysfunction and epigenetic dysregulation, and they have become targets for interventions. And it's important to note that if you could cure cancer or heart disease in a 50-year-old woman, because of the amount of wear and tear she has already accumulated, we would only add maybe 2 to 3 years of healthy life expectancy to her before she had another disease. If you could slow the aging process, as we speculate within the geroscience hypothesis, then you could add up to 25 years of healthy living. The economic value of this is also quite staggering. Like currently, if you draw a very crude analogy, think of our body and its component parts as being like a car. Our medicine and our science is very good at telling us how to replace bits that go wrong and keep the car on the road. What it cannot do is deal with this when the car is put in the wrong environment and it gets trashed. This is the effect of your exposome. So the exposome comprises the totality of your environmental exposures. That's both biotic and abiotic, preconception to death. And it can be broken into a number of different layers over your life course. And it's for molecular biologists and certainly for pharmacologists, this is very woolly stuff, but it has impact because how you're treated as a child by your parents, it's important to your adult health. Are they abusive or are they supporting? Are your siblings abusive and supporting? Is the environment you live in conducive to your safety? Do you get a chance to be well educated, to exercise? Are you living in a toxic and polluted environment? Do you have good transport? Do you have good social circles? And of course, the one thing that we find from the most long-lived and healthy communities on the planet is there a degree of social connection, often manifest in spirituality. The importance of all this can be highlighted by the fact that 3 simple exposome factors, air pollution, tobacco smoke, and diet, account for 50% of global mortality annually. So we need to get a grip on how our genome and epigenome is interplaying with our environment across our life course, because this is a major determinant of how we age. Back to Hippocrates again, diet, all diseases begin in the gut. In Glasgow, if you look at what drives accelerated aging, we see that an imbalanced diet, not eating enough fruit and vegetables, is tied to inflammatory status and aging, biological aging, as measured by a range of different markers. It is also associated with epigenetic differences in the microbiota, and psychological stress, the sequelae of which is often a range of different burden of lifestyle diseases. And again, linked to diet, we see those that eat too much red meat tend to be hyperphosphatemic and have mild to moderate chronic kidney disease. So these are people in the general population, not a clinical population. Interestingly, the biological aging component of these individuals is not good at explaining their inflammatory burden. Intuitively, the more old cells you have, the greater degree of inflammation you should exhibit, because senescent cells have a pro-inflammatory secretor. However, in the general population, despite what's been observed in tight clinical and laboratory conditions, or under those conditions, in the general population, only about 10% of the inflammatory burden appears to be explained by biological age. So the majority of it is coming from somewhere else. The other thing to point out here relative to diet and aging is that serum phosphate, so it's nutritionally acquired phosphate, the levels are strongly correlated in mammals with longevity. So if you look at the Clotho mouse here that lives 12 weeks, he's highly phosphatemic. Human centenarians have got low levels of phosphate. Most of us have slightly higher levels, and human progeric mutants, such as those with Hutchinson-Gilford's progeria, exhibit higher levels still. This is a malleable component of your exposome that you can use to reduce the rate at which you age through diet. We have been exploring this in the context of kidney disease, again linked to Glasgow, for a number of reasons. One, because chronic kidney disease is a disease exhibiting features of accelerated aging, both cellular and molecular features, and also physiological features. So you've got muscle wasting, osteoporosis, vascular calcification, cardiovascular hypertrophy, alongside the diminution of NRF2 and standard cytoprotective responses. Interestingly, if you compare a range of different multiple organ chronic diseases and mortality, the most extreme in terms of a difference between your biology and your chronology in aging is chronic kidney disease, where the difference is about 5 years. So it is a good system for teasing out subtle differences. Typically, if you go back to the hallmarks of aging, we have shown quite consistently that we can find all of them in the context of chronic kidney disease. And we want to exploit them because if you have chronic kidney disease and you're on dialysis, your survival time on dialysis is about 50% over 3 years, and you really need a transplant. But only 1 in 7 patients waiting for the transplant gets it. And the transplant will make you physiologically more robust, but we need to know if it's slowing down aging. It's certainly not reversing it. And to do that, we've had to develop biomarkers that can actively and accurately report on the efficacy of any interventions to slow down aging. So these are xenotherapeutics. The biomarkers need to tell you accurately not what's happening when you have a disease, but what's happening before one develops, when you're starting to move off your normal trajectory of aging. So you need a biomarker that will tell you how many miles you have traveled in your life course, so acting as an odometer, and then one that's telling you at what rate you're moving at, so a speedometer. There are a range of these out there, and we've got many sophisticated cellular and molecular markers. We use telomere length, CDKN2A expression, advanced glycation end products as part of skin fluorescence measures, phenotypic age, which correlates a range of blood markers. We've got proteomic markers. We've even got microbial metabolomic markers, but most of them are not particularly accurate at predicting how far you've traveled or what rate you've traveled at when you haven't got a disease. And still, our best markers have been biophysical, so gait speed or lens density in the eye. Just to exemplify this, skin autofluorescence and phenotypic age when you have chronic kidney disease give you biologically implausible measures of age. So if you're chronologically age 60, SAF can say that you're biologically 200. That's just not good sense. It's an inaccurate marker. And again, phenotypic age tells you you're over 100 when you're 20. This is just not accurate. And these are actually sold as commercial tests as well as used clinically. They are not accurate. We've tried to move away from this and look at epigenetic clocks based on changes in DMA and methylation. These are seriously good tools. They're clever stuff, and originally developed by Steve Horvath and his lab alumni. They accurately track chronological age, and they are good at showing you a disconnect between chronological age and biological age when you have a disease. They have really needed to be tested clinically to see if they report accurately on what most of us want to know, physiological function. And we've tried to compare them head to head in a renal transplant setting where we know an individual with chronic kidney disease shows accelerated aging. And we're asking, relative to getting a transplant and following that transplant for a year and looking at the improvement in kidney function and the clearance of toxins from the body, just what this does to the individual's age when you start looking at their blood and measuring their epigenetic age. So typically all the clocks, 4 of these were published in comparison, and GrimAge, which we got very kindly as an algorithm from Steve Horvath, we haven't published with yet, but they show the same thing. Good correlation with chronological age when you have chronic kidney disease. If, however, you have a transplant and you follow that transplant for 1 year, what we observe is they are not good at tracking the rate of aging. GrimAge, the Hannum clock, and the Horvath clock do not work well in this context. PhenoAge, developed by Morgan Levine, is another great tool. And again, it does track, but it is not particularly robust. And we're trying to improve on this at the moment. But a composite clock that we have developed called the Galacto-Karolinska clock tracks accurately. What we show is that physiologically, your renal transplant makes you better, and it slows the rate at which you're aging, so the rate at which you're accumulating miles in the clock. And it does so in a clinical setting. So having that, it gives us an accurate tool to start evaluating any other interventions designed to slow down aging, for example, with NRF2 activation or other strategies that are being developed pharmacologically. Typically, what we show here then is that CKD patients, again, show that 5-year age difference between onset of disease and chronological age, and that will only increase as disease progresses. But when they get a transplant, it slows it back down to the normal rate of aging. It does not reverse the aging process. Another feature of this that's important with respect to environment is keeping methylation on your DNA and keeping your epigenome stable. And what we observe in our Glasgow population that is most biologically aged is that we have telomere attrition, hyperphosphatemia, more inflammation, and this imbalanced diet, poor renal function. The imbalanced diet allows, through eating too much red meat, the generation of trimethylamine, which is converted from the breakdown of carnitine by bugs in your gut, and then onwards in your liver to generate TMAO, which is highly inflammatory. On the same biochemical pathway and on a different branch, if you do not have the right bacteria in your gut, you cannot generate sufficient levels of betaine. And betaine is a main methyl donor that feeds into the SAM cycle and helps maintain your methylome. And consequently, when we compare the most and least aged in the general population in our city, we see that they have the most aged, again, have less methylation on their DNA. And we've asked whether or not they have a dysbiotic gut. And this is what we find. We find that in general, they have more oral pathobionts detectable in their system and a loss of health promoting bacteria or salutobionts. And these features are linked to faster aging and poorer health. Unsurprisingly, when you have a bad diet, which is typically very Western and full of ultra-processed foods, you do not have a system that is good at promoting the growth of salutobionts. Typically, we have lost the pickling and fermentation processes to preserve food that are observed in traditional diets that allow these bugs to thrive. They're able to break down phenolic acids in fruit and vegetables that our deprived do not eat, and they can generate alkyl catechol that can activate NRF2. So TMA, generated from eating too much red meat, is now been shown to be prominent in promoting atherosclerosis, chronic kidney disease, rheumatoid arthritis. Not a pleasant situation, but again, one, as part of the foodome, which is part of your exposome, you can manipulate. We've also asked what happens in a transplant setting and where you slow down the rate of aging. We've asked, can you actually restore a normative gut microbiome? And the answer is we can improve the microbiome and generate more salutogenic bacteria, but we never return it to a fully normative setting. And interestingly, this also tells us that the bugs that remain present can still generate TMA, and that TMA level explains up to 25% of inflammatory burden. Two metabolites, so TMA and polyacetylglutamine, contribute to almost 40% of the inflammatory burden. And these are things that are normally removed in dialysis. So again, this opens up a route to reducing inflammatory burden and maintaining better health in old age and slowing down the rate at which you're aging by modulating the microbiome through food. So this is something we've been exploring, trying to provide diets that will both directly support the growth of salutogenic bacteria and indirectly support activation of NRF2. This approach has received some support from a recent study showing that life expectancy can be increased by up to 10 years following a shift towards a healthier diet in the UK. So cutting out red meat from domestic ruminants and increasing particularly the amount of fruit you eat. And we've taken this on board and clinically tested the approach of using prebiotics. So this is using starch cookies containing inulin that support the growth of salutogenic bacteria that can produce activators of NRF2. They can also reduce inflammatory burden because they produce short-chain fatty acids. And we've tried with 4-week interventions in hemodialysis patients, which show accelerated aging and increased inflammatory burden. And what we find, in short, is that that simple intervention is able to make the individuals more robust. So we're really pleased with this. And we've now gone to the extent of completing a randomized controlled double-blind trial using a range of polyphenolic agents that can also activate NRF2, and in particular, propolis. Propolis, like many of these nutritional interventions, is not particularly desirable all the time because its formulation is never consistent, and it depends on the region where the bees are foraging to make their nectar, or honey, and to make their honeycomb. And propolis is a mix of polyphenols that are part of the wax compartment of that honeycomb. Another thing that can be done here is direct microbiome transplants, and recently fecal microbial transfer between young and aged mice has been shown to reverse the hallmarks of aging, both in the gut, eye, and brain. So this is a really promising area for exploitation in trying to slow down aging and improve health span. So I will stop and basically tell you that the exposome is a powerful mediator of aging and health, that it is amenable to simple interventions, often focused towards manipulating the activity of NRF2. And critically, we now have tools to assess these for efficacy in real time, because human aging is very gradual and heterogeneous. We live long lives relative to our size, and we need good tools to tell us exactly what is happening. So hopefully, these are things that you may think about using as your pharmacological interventions develop in the coming years. Thank you. I'll finish with more Hippocrates saying, if we can give everyone the right amount of nourishment and exercise, we would have found the safest way to health. A 3,000-year-old message, but still pertinent today. And I'll just thank my group and in particular my collaborators at the Karolinska Institute, led by Peter Stenvinkel, Maastricht Leon Skurgers, Rick Johnson in Colorado, and Colin Selman here in Glasgow. Thank you.
Thank you so much, Paul. Lots of questions. I want to know how to stay young, hopefully,
Don't we all?
for later. And now I leave the word, the presentation to Brigitta.
We need to live longer, to live better. We have to make a lot of things. We have to eat answered food, quindi modify our microbiome and our lifestyle. So, okay, now we can go on. And the question at the end of the talk. It's a great pleasure to introduce the last speaker of this webinar series dedicated to the oxidative stress and aging, who is Professor Ioannis Trougakos. Ioannis is the head of the aging and age-related diseases group at the National Kapodistrias University of Athens in Greece, but he's also a working group leader in bed-by-far construction, a dedicated time to the economic exploitation. And he has a proven track record in aging research by using a multidisciplinary system biology. And thank you very much, Ioannis, to be here. And the title is a presentation is Proteome and Redox instability as a major driver and the hallmarks of aging. So the floor is yours. Thank you, Ioannis.
Okay, thank you so much, Brigitte, and of course, many thanks to the organizers for the opportunity participating in this so very interesting webinar. So as said, my title would be Proteome Redox Stability as major drivers and hallmarks of aging. And just to start with a very short description of what we are trying to do in my lab. So we are an age-related diseases group. So we are applying a systems biology approach, aiming to understand the different levels of regulation and crosstalk of the various proteostatic machineries, how these proteostatic machineries and the proteostasis network is being functionally integrated with mitostasis and genomic integrity. The systemic effects which are reduced by the specific laws of proteostasis, and of course, which is of significant interest, as was already highlighted by the previous speakers, what's going on regarding the deregulation of proteostatic modules in age and age-related diseases. So we're using different models, including mammalian cells, Drosophila flies, transgenic mice, who have a quite strong translational continuum, with many collaborations with clinicians as we are participating in trials in the clinic, and also we as well will present during close to the end of this lecture. We are having a screening platform where we screen natural compounds, small molecules, etc., in order to found molecules that can maybe increase longevity and, more importantly, healthy aging. So this is the structure of my lecture. So I'm going to describe briefly the major architecture of biological systems regarding proteome stability and endocrine regulation. I will present the molecular biology of aging, and also I will just discuss very briefly interventions that can maybe prolong healthy aging. And just to mention that for this lecture, I will prioritize concepts over experiments. So this is a typical biological system, which is extremely complicated, because when we speak about humans, then we have a system with more than 35 trillion cells, 200 different types, and then we have the idea of small systems like the mitochondria and all the different organs that we found in the cell. So you see here that we have the idea of integration. This is also the case when we see the different systems, and all these are being made for protection, energy, and reproduction. Now, when it comes to the cellular level, this is the basic design that we see in all biosystems. So there are some chemistries, nucleic acid, polymers, which store the information. Then you have the word of function, which is the proteome, basically, and the entry point is the protein synthesis. We have the chaperone that basically folds and curates all these small polypeptides. And then we have a number of degradation systems if needed, and some of these peptides cannot be folded properly. These can be targeted for degradation either in the ubiquitin protein system pathway or in the autophagy lysosome pathway, where you have degradation of much more bigger assemblies like protein aggregates, mitochondria, etc. And it's always a decision about fold, or degrade. Of course, for these processes, you need huge amounts of energy. This being, among others, provided by mitochondria. And for the system to perform well, you need a number of sensors. I will also discuss NRF2, and all these sensors are basic transcription factors. They are short-lived proteins, so normally NRF2 and all these others transcription factors like 53, Hitchcock factor 1, FOX, etc. They share the same properties. They are short-lived. So in this case, NRF2, this has been already introduced. It's been bound to KEAP1. This enables choline 3 to bind also, ubiquitination, and then the protein has been targeted to potassium for degradation. In case of any challenge, say for oxidants or electrophiles, then KEAP1 is oxidized. The complex cannot be stabilized. NRF2 is free, enters the nucleus, and by binding to antioxidant response elements, drive the expression of, say, antioxidant enzymes, and as we'll see, many more protective molecules. So this is a more close, if you wish, view of the proteostasis network. So this is again the entry point, which ends up with millions of these small Legos, polypeptides, which have to be recognized, folded, and then sorted, of course, and then transferred to the different parts of the places of the cell where they are functional, where you have the assembly process, and then follows the functional integration, which makes this cell to be operational. And of course, you have the degradation pathways, which function in different pathways, including the protein synthesis quality control. So, of course, as I said, you need energy, which has been it's been produced as part of the catabolic and anabolic pathways. Almost the entire energetics are being produced in mitochondria. It's really impressive that the human body turns over its weight in ATP daily, because we have 1,000 to 2,000 mitochondria per cell in all of these more than 35 trillion cells in our body. This is the oxidative phosphorylation machinery, and ATP is being produced by nutrients and oxygen, which is absolutely needed for these machineries to perform their main task, which is the production of ATP. Part of this process is also the production of these reactive oxygen species, which include different types of molecules which are highly reactive, including superoxide anions, hydroxyl radicals, etc. These are considered as free radicals, but also we have ROS, which have oxidative properties but are not free radicals, such as hydrogen peroxide, etc. Now, of course, just a brief introduction about the sensor. Again, this has been also discussed. So this is the NRF2 KEAP1 system. NRF2 binds to KEAP1 in the absence of any challenge. It's been ubiquitinated and sent for degradation of the proteasome. In the case of oxidative or electrophilic stress, KEAP1 cannot bind NRF2, which then, as said, localizes the nucleus and drives the expression by binding to antioxidant response elements. Interestingly enough, this sensor is also regulated by growth factor signaling. Especially, it seems to be downstream to the insulin PI3 kinase AKT signaling. And when this PI3 kinase AKT signaling phosphorylates glycogen synthase kinase 3, which is an inhibitor of NRF2, this part of the pathway cannot be operational. Again, therefore, there is no ubiquitination of NRF2 by the complex of beta-TRCP and QL1. Then this is being free, translocated again the nucleus and drives the genomic responses. So you see that these type of sensors, they are sensitive to different types of signals. And just to mention, I don't have time to enter into details, that this NRF2 pathway is being modulating many genomic responses, including mitochondrial genes, autophagic genes, etc. So this is how it goes when we start to think about aging. Why? Because all these pathways that have been describing work very efficiently up to this point, which is exactly what is happening in the nature. And then we have this period of life, which is highly unnatural, okay, and doesn't exist actually in nature. And this is the period of aging, which is a condition which is marked by reduced chance for survival and health. And the biological system is very disease-prone. This is a very disease-prone state, and of course leads to death. And just to mention that aging is, of course, an inevitable consequence for life for nearly all organisms. But again, it's been seen mostly in humans and animals in captivity. Of course, I want to state that no cell type is spared from aging. So it's a process which we don't frequently or never see in nature. And also it's highly stochastic. Why? Because there is no any evolutionary programming behind this process. This is also shown here, where it's epidemiological data from UK. You see that essentially all of these major age-related diseases, like cardiovascular diseases, cancer, and dementia, they start to appear late in life, and therefore they're clearly age-related diseases. So these are the hallmarks, as we call them, of aging. You see many different pathways which are deregulated during this process, including inflammation. This was very nicely introduced already by Paul, but I will focus on this one, which is mitochondrial dysfunction, because this relates to increased levels of ROS and also to loss of proteostasis, meaning that the proteasome the protein becomes dysfunctional, including disabled macroautophagy, meaning that also the processes of removal of aggregations, dysfunctional mitochondria also become dysfunctional. Now, we and others have been introducing the idea that the loss of proteostasis, meaning that an increase in proteome stability, is like the driving force of aging of most, if not all, age-related diseases, because the decline of a core PN component, like for instance, the NRF2 pathway or the ubiquitin proteasome pathway, or the suppression of the signal's competence during physiological aging saturates this pathway, which then triggers the collapse in genome stability, proteostasis, mitochondrial functionality below a threshold that we know that in the young organism sustains cell viability. Now, this is what's happening in the proteome in a young cell, or where in a young organism. So you have the polypeptides, chaperones take care of those, and eventually folding intermediates or partially folded states, and then folded to native states. Now, when the proteome becomes unstable, or there is a accumulation of ROS, oxidative stress, etc., these pathways become less and less efficient, and therefore you start to have amorphous aggregates, oligomers, and essentially and eventually amyloids, which is a major trigger for neurodegenerative diseases. Now, this was quite interesting because it was also found that by the group of Cynthia Kenyon that protein aggregation, it seems to be an inherent part of aging in C. elegans. So it means that likely this pathway is something that's taking place during aging, not only in humans, but also in most of the higher metazoans. This is data from our lab in the fly model. So this is the proteasome activity during aging. So you see that you have a down-regulation of proteasomal genes, including expression of protein subunits, of proteasome subunits, sorry. And this also shows that we have a reduced effectiveness in the assembly of this protein machine. This is an immunoprecipitation experiment where we have been immunoprecipitating with A subunits and probing with a beta-5 catalytic subunit. You see that there is a reduced assembly of the proteasome in aged flies, and this was also nicely associated with reduced activity of the proteasome during aging. So something that is not clear nonetheless in relation to this type of loss of proteome functionality, of proteasome functionality, is whether this takes place in a linear fashion across different tissues, whether it's been starting in a later age. This introduced the, let's say, the provocative idea that maybe there is some program behind the collapse of proteostasis. And of course, we don't know whether this is the rate of this loss of proteostasis is similar across tissues. Now, then we asked, okay, what could be the case if we take a young Drosophila and we inhibit proteasome to the level we see in the old flies? So this is the experiment here. So in this case, we have been doing this type of experiments, of course, with genetic means, but also with proteasome inhibitors. Here we are using bortezomib. Bortezomib is a drug, a very specific proteasome inhibitor used in the clinic for multiple myeloma treatment. You see here we have been using increasing concentrations of this proteasome inhibitor, and you see that nicely dose-dependently inhibits the proteasome activity. You see an increase in ubiquitination and oxidized proteome. And what is really nice is that you see an dose-dependent decrease in neuromuscular activity, and of course a very much dose-dependent decrease in life expectancy, meaning that if you introduce proteotoxic stress in the young fly, then you accelerate aging significantly. Of course, this correlates with a significant accumulation of oxidative stress in the fly's tissue, including both ROS and hydrogen peroxide. And we know this is, and we have found that this is happening because when you disrupt the activity of either ubiquitin proteasome or autophagy lysosome pathways, then also gradually you start to lose your mitochondria, because mitochondria and mitochondrial functionality very much depends on the activity of this proteasome, ubiquitin proteasome, and autophagy lysosome pathways. And I don't have time to enter into details because we have been also seeing what's happening when you disrupt mitochondria in these systems. It's also the case that when you start to disrupt mitochondria function, gradually you lose proteome activity, or proteome becomes unstable, meaning that you have a system which is in balance, and then all these systems have to perform effectively or efficiently for us to be young. Okay, this is modulators and targets of ROS. You see that the main source relates to mitochondria. Okay, and you see the sensors. This is the data from C. elegans, the sensors that respond to this accumulation of these oxidants. And apart from what's happening, of course, in the cell, like the mitochondria, NADPH, oxidase, inflammation, of course, to have the exposome, all these insults from the environment, and this was also nicely discussed by Paul. So there are different sources. Luckily, you don't see any accumulation of ROS, whereas when we're young, or this is controlled as part of the effective functionality of the sensors. Actually, we start to see the accumulation of ROS as we get older. So this is an experiment, again from the fly model. In this case, these flies were living very happily in the fly room, so there is no any actual exposome as such. And you see that as they become older, they start to accumulate ROS, including hydrogen peroxide. And okay, we're now working on that. The rate of ROS accumulation is much more significant in males versus females. This is accompanied by a higher accumulation of DNPs, meaning that the proteome is getting more and more oxidized. And when it comes to all these metabolizers, including also mammals, this accumulation of ROS relates to different phenotypes, including cellular senescence. You see ROS here, which also relate to increased inflammation because of the secretory senescence-associated secretory phenotype, the SASP, and of course, this state of increased oxidation correlates with most, maybe all, age-related diseases, including neurodegeneration, osteoarthritis, reproductive diseases, cancer, etc. Now, how about the sensors? We have also found, again, in the fly model that sensors become dysfunctional as the flies age. This is the case of NRF2. This is a transgenic fly where the GFP is downstream of the antioxidant response element, and this is the mutant. Okay, so let's focus in this part of the slide. So this is the actual values. This is the relative GFP fluorescence. And in this case, we challenge the flies with a proteasome inhibitor because we know that if you suppress proteasome activity, then you get mitochondria deregulation and an increase in ROS. You see how effectively NRF2 is being activated in the young biological system and how poorly this is happening in the old flies. Okay, and again, what would be the case if you take a young fly and knock down NRF2? You see here the knockdown. This decreases the expression of the proteasomal subunits, less proteasome activity, accumulation of ubiquitinated proteins, meaning that the proteome also becomes quite unstable. And of course, these flies die earlier as compared to the controls. Now, these findings at that time brings us quite some hope that if we overexpress the sensor, then we could maybe able to increase longevity, right? We have been trying again in the fly model, but also in many other systems to activate NRF2. And it was the case that whereas sustained NRF2 activation extends indeed health and lifespan, this was not the case when NRF2 was activated for long and high levels. And this was a paradox because these flies were very tolerant to stress, but they were aging faster. Why? Because there was a metabolic deregulation. And this is the reason, because as part of the network of the NRF2 activation is that the NRF2 also targets its inhibitors. Why? Because this is how the system works, because this is a short-lived protein and it has to be active when there is a challenge. So it starts to activate cytoprotective proteins and genes, but at the same time, it also activates its inhibitors. Parts of the transcription targets is this insulin growth factor inhibitor. So this causes a significant, I don't have time to enter into details now, but this is a significant metabolic deregulation resulting in a premature aging. So this has teach us that regarding the activation of these stress sensors, which I said again in mild levels or low levels, if you wish, it's really beneficial. There are many things that we have to control, if you wish, including when, where, how long, how much, in which tissue, etc. So we have been studying more regarding the activation of these proteostatic modules in different models of neurodegeneration, humanized flies with expressing Parkin and PIC1 genes, models of Alzheimer's disease, or even models where we have been introducing cardiac toxicity because of proteasome inhibition. And as you can see in these papers that I cite here, in most cases, actually in all cases here, we have been shown that mild activation of proteostatic modules has beneficial effects in age-related diseases. So this brings me to the last part of this lecture. And okay, so this seems to to bring some hope that we can maybe increase health span, and as also discussed by Paul, this is quite important because we need to increase healthy lifespan. So what have we doing up to now is that we're trying to develop drugs in an aged tissue when the disease is already there. The idea would be maybe to prevent the appearance of these diseases. How? Because this is the only way that we can do that in humans, by using diet, where by exploiting natural products, we can maybe increase health span and therefore suppress all these diseases. So we have been setting up in the lab quite, I think, effective screening platform where we screen natural products, small molecules, etc. These are different EU grants who have been participating, screening natural products from marine organisms. Here it's from microorganisms, Mediterranean diet, etc. And we have now a portfolio of natural compounds, extracts, or small molecules which are quite effective in activating these proteostatic modules and show some pro-longevity effects in different models. Just to show this example, in this case, we have been working with this 6-bio molecule. This was isolated from this mollusk, and then following a chemisynthetic modification, we found that this inhibitor of GSK3 here, which, as I said, is a suppressor of NRF2. For instance, in the fly model, we found that administration of the 6-bio delays age-related phenotypes like the loss of locomotor activity, increases resistance to sensors, decrease glucose lipids in the hemolymph, and more importantly, it seems to increase health span, but also lifespan. This is happening. I don't have time to enter details because it by suppressing GSK3 mildly activates NRF2. This 6-bio molecule also seems to suppress PDPK1 kinase, so it's sort of a mild caloric restriction mimetic. So we're still developing this molecule because we think that can be really effective also in mammals. Why that? Because we have also found that in normal human diploid fibroblasts, it suppresses cellular senescence mediated by molecular damage. How? By activating cytoprotective cellular modules. Okay, so I would like to conclude with this presentation by acknowledging all the members of the lab, and including the previous ones, for being very effective through years in these projects. Of course, many collaborators have been contributed in these efforts. For this lecture specifically, I would like to thank Dick Borkman from Rochester, US, and especially Jerry Sikiotis from Lausanne. Jerry also is also a member of our Ben-Bed FAR construction, with whom we have a very fruitful collaboration through years. I'd like also to thank collaborators from the clinical therapeutics department in our medical school, Professor Dimopoulos, Kastritis, Amatolopoulos, and Terpos, with whom we have a very fruitful collaboration through years in multiple myeloma, which is a paradigm of increased protoxic stress, which leads to cancer. And now have been starting working on TTR amyloidosis. Of course, all of our founders, including constructors, and in particular, Ben-Bed FAR and Professor Antonio Quadrado, for being so effective in coordinating this construction. And I will leave you with this motto of the lab: okay, it's nice to add years to life, but maybe it's more important to add life to years. Thank you so much, and I'm happy to take any questions.
Thank you so much, all the speakers. Thank you, Ioannis, for these interesting talks. And now is the time for the question and answer. And we received some questions, and the first is from Roberta. So, Roberta, you can.
Yes, I wanted to. I had several questions. I wanted to start with Anna.
Okay.
In relation to your S-nitrosylation of KEAP1, if I understood correctly. So KEAP1 is nitrosylated, S-nitrosylated. And then you showed with your pull-down assay that it's bound to, now I don't remember the other proteins, but you're saying basically that NO is moving from KEAP1 to these other proteins, affecting, this is what you're suggesting, right?
Mm-hmm.
So that was my first, to make sure that how is this movement occurring? How is the NO moving? And the second question is if you tried nitric oxide synthase inhibitors to see whether there is a change in the S-nitrosylation, but also a change in the senescence of the NRF2 deficient endothelial cells.
Okay, thank you for those questions. They are really interesting, and maybe I can start with the second first, at first, because we haven't tried to inhibit NO production. We only tried to knock down iNOS and eNOS, and in both cases we observed a strong decrease of S-nitrosation. It was stronger in case of the iNOS knockdown comparing to eNOS, but in both cases there was the decrease is S-nitrosation. We haven't tried, for example, with L9. And regarding the question how KEAP1 works in a way that it facilitates S-nitrosation, this is really very interesting thing, because actually, if I may comment a bit more, when we did those experiments, a paper was published that was done in bacteria, and it on S-nitrosation, and this paper showed that in bacteria there is a very similar complex catalyzing S-nitrosation, consisting of GAPDH, so the same as in case of mammalian cells, NO synthase, which in bacteria it is NAR-GHI protein, and the protein that is the SNO synthase, let's say, in bacteria, this is HCP, that is directly, this is a direct target for OxyR. And the OxyR is the major transcription factor that orchestrates the genes involved in an oxidative defense system in bacteria. So it seems that this is really very, that those complexes between a bacteria and mammalian cells are very similar. So that made us, let's say, more, I think that in this view, our conclusions on the KEAP1 and SNO are even more are stronger simply. And regarding the KEAP1 itself, so KEAP1 can be S-nitrosated, and at the moment, actually, we are mostly limited by the, let's say, not understanding how S-nitrosation occurs, but this is highly likely that KEAP1 is the SNO synthase. However, taking into account that NO must be oxidized to form NO plus to react with thiols, and zinc that is bound in KEAP1, it cannot achieve that. So our first hypothesis is that either zinc can be replaced by, for example, ferrous iron it could bind to cysteine in KEAP1 and catalyze this reaction, because it is stronger. let's say, transition metal that would be able to oxidize NO to NO plus cation, or another thing may happen. And our second hypothesis is that because we observed the increase in the superoxide anion in those cells, and it may happen that if the KEAP1 molecule is in a close proximity to superoxide anion, then the reaction between those two and the cysteines of KEAP1 may get into the reaction with superoxide anion, forming the intermediate that would finally react with the NO, ending up in the S-nitrosation of KEAP1. And then we believe that the transnitrosation of the other proteins can be achieved by GAPDH, as it has been already proven. So that would be two mechanisms that we are considering. However, we have no experimental proof for that. But I think that it is rather from the experimental point of view, it would require more, let's say, sophisticated methods that are not used in our lab. So
of course, it's difficult
let's say
to
chemical
demonstrate.
ones, or
yes,
yeah. But
yes.
anyway, this is really interesting, I believe.
Yeah. Yeah, maybe did you check also glutathione levels, the levels compared, because
yes, we have compared the level of glutathiones, the total and the oxidized and the reduced one, and the total antioxidant capacity is a bit lower in case of NRF2 deficient cells, and however, the glutathione levels are, maybe they are affected a bit, but this is not like a very strong effect.
okay, thank you. Very biochemical.
Yeah, it is very biochemical.
Yes.
Yeah. Thank you.
I have a very good question for Anna, if it's possible.
Yes.
Anna, it is known, or it has been shown, that also NRF2 overexpression can induce premature senescence. So have you tried to see whether this also impacts on MIR34A expression, functionality, activity, whatever?
So we haven't tried with the NRF2 overexpression. We have tried with the NRF2 dominant negative overexpression, and in this case, the cells were not affected unless exposed to turbulence, right, to oscillatory stress. And we have tried with the overexpression of KEAP1, and actually if I remember well, in terms of S-nitrosation, there was no difference. However, we observed, for example, increased formation of protein aggregates and the loss of proteostasis with the overexpression of KEAP1. So, but in terms of when we did the overexpression of NRF2, the regular one, the normal NRF2 with adenoviral vectors, actually, for example, we observed that the cells are more prone for migration. We observed the increase in the angiogenic response. So, but we haven't observed the senescent phenotype.
But you mean this was done in Hubeck cells, in normal diploid?
In human aortic endothelial cells.
Okay. Because there were some papers previously showing that NRF2 overexpression more or less behaves like oncogene expression, right? To use premature aging. Yeah. Anyway, thanks.
Yeah. So we haven't observed that for the aortic cells.
Mm-hmm. Great. Thank you.
Thank you.
Any more questions for Anna?
I write a general question for all speakers. In the light of the signaling pathways that show that KEAP1 and NRF2 axis play a prominent role, what compound as NRF2 activator could be used for reducing or preventing aging? We can start to think to use this compound to prevent aging. Quindi should it also possible turn to drugs that have already been recognized as safe to treat aging? So about drug and that can target NERV2 axis.
If I could say something in that there are many, many drugs currently in clinical trial. I think there are around 350 ongoing trials with putative xenotherapeutic agents, many of which show some degree of interaction with NRF2. I think part of the problem is identifying when and where in the life course these drugs can be used and for how long. There are also a range of repurposed, clinically approved agents that are currently being exploited in this space. Many of them still have undesirable effects outside the area of xenotherapy. So metformin and rapamycin are currently used clinically. They have, certainly in the case of metformin, a degree of nephrotoxicity and hepatotoxicity. So dosing critical. I don't know personally if I would take rapamycin or metformin. They were developed for a specific purpose, and they may not affect all aging cells, given that many different types of senescent cells reach senescence by different means, so different pathways, and there may not be a one-size-fits-all approach. We're really still in our infancy with this. We don't understand aging completely. We have a very good understanding of the mechanistic basis of cellular effects, but we don't have a holistic picture of where everything fits together and whether or not you can intervene early in life or later in life appropriately. We do need, for example, senescent cells when we're old, not when we're young, to reduce physiological capacity. But when we're older, they are oncoprotective. We do need them for wound healing. So there's a balance needed. I think there's a gold rush currently where people think that they can extend lifespan. And industry certainly wants small molecules that can do that. I think that's unrealistic. I think we can improve our years of healthy living, but even if you look at current experiments in mice and Drosophila and in xenohabditis, if you have good xenotherapies that are extending the lifespan of those model organisms, they still die. They don't live forever. And I think we just have to have a more realistic appraisal of this. And despite, as I'll repeat, the molecular and cellular complexity that we have developed an understanding of, the best approach is to slowing the rate of travel, so slowing the rate at which you age, or well-established balanced diet, exercise, being social. We always like to think that we can extrapolate our primary science from cells and small organisms to a complex human and the complexity of a human society. It's not easy. And we should, at least if you use the old Occam's razor, simplest explanation is always correct. Stick to the things we know with best, and at least then use those as a platform to develop more comprehensive science. And you've seen from Yanis's presentation, there are lots out there that we really still need to understand. The pharmacological interactions and the molecular and cellular interactions to exploit in this context. If we're going to live for longer with, and use the drugs we have, my guess is that we will have to replace parts. We'll have to grow them and transplant them or reprogram to regrow. We're not, going to get rid of the wear and tear that we have accumulated over the course of our life. We can just slow down the rate at which it arrives.
If I could add a very quick comment on what Paul said, I would say I fully agree. Coming to the compound, NRF2 activator compound, whatever issue, I would say there are many, many compounds that can activate NRF2. It can be activated either by disrupting the KEAP1 NRF2 binding or by modulating the metabolic pathways, GSK3, etc. activity. I would go for a mild activator, but again, as I think very nicely pointed out by Paul, and I fully agree, we are not in the phase of one size fits all, A. B, the system is, I mean, not the humans. Drosophila is extremely complex. We see that if we overexpress NRF2 in the neurosystem, then you see different responses when you do the overexpression in the muscles, right?
Mm-hmm.
So there are so many things that we don't know. We don't really understand how exactly the different systems communicate, right? Because we know that if we disrupt proteasome activity in the muscles, then gradually you have the cell non-autonomous effect where the other systems start to collapse. We don't know how this messaging is taking place. So it's extremely complex. We need to accumulate much more knowledge. I would agree with being social, having a Mediterranean diet, having good sleep, and being in a good mood as much as possible with some but not much stress. And but on the other hand, we need funding. We need to to accelerate our research on these topics. Why? Because at least in the Western countries, we are facing a significant aging population. This means that this will increase also the pressures in the insurance systems, in the health system. Why? Because more and more people will ask, medical care. Why? Because we're going to have a significant increase of these age-related diseases. And a final very good comment is that, NRF2 is, activation of NRF2 may, be beneficial in terms of increasing, prolonging the health span and also protecting us from neurodegenerative diseases. But don't forget that we see NRF2 overexpression, proteasome overexpression in cancers. So it's not so straightforward, right?
But Ioannis, has anybody tried to give like an inducer of NRF2 transiently? I give it every 3-4 days, for example, in Drosophila. I don't know if it's possible. Because we know that the genetic manipulation is one thing, which is very different, doing mild activation, which is there over time. Whereas, if I think about the system of NRF2, which is supposed to respond to stress, but then it's supposed to go down and become, we don't want to have NRF2 all the time. So I'm thinking about an approach where we transiently increase every few days, once a month. I don't know exactly what would be the protocol, but has anybody tried that?
Actually, we have been trying that because we have been trying many different approaches so that we, because the thing is that at least in the fly model, because we're doing things in mice clinic, also in the cells, in human cells, the flies, there is actually minimal genetic redundancy, so we don't have different NRF2 isoforms, for instance, or including all the other FOXO, Hitchcock factor, etc. So it's much more convenient because you have a very simpler, biological architecture. Okay, that's good. We have been trying that too. I can tell you, Roberta, we have very powerful data regarding health span increase, longevity, unpublished data. From this point up to the point where something can be tested as a clinical trial, I think we have many things to do. It was really happy. And it was very interesting for me that there was a Nature Medicine review lately about how diet, people start to think seriously about diet, because the only way to modulate health span in humans, because we cannot do genetics, you cannot do caloric restriction, this is impossible with the population. The only way is to suggest, educate the population about the significance of diet, including all the others that have been also introduced by Paul. So diet is critical. We need to get some more knowledge on that. The approach of, oscillating NRF2 activation, this is something that we and others have been testing. It seems to work, but it's a different thing trying something in the mouse on the fly, but as compared to humans. Yeah.
I want only add something. In the Italian population, other to the Mediterranean diet, only the 5% of the population other to Mediterranean diet. So it's very difficult to. We have to rewrite the healthy diet, because people now use industrial food. Quindi, so it's difficult to prepare a diet for all the people. It's
I fully agree with that.
Okay. There is something around the Mediterranean diet that's important to mention here. There is good evidence, and it's published, that if you factor in deprivation status, the protective effect of the diet is lost. So it's only the people that are socially more affluent that get the benefit. So it's one of the things I was saying earlier. Often a lot of the current evidence is oversold, and we tend to look at human studies that are not very well powered, or that they have not factored in confounders such as psychosocial factors or socioeconomic factors. And I was delighted when you said that such a small percentage of the Italian population has that diet, because I think most people will automatically think everyone in Southern Europe has a superior diet, when they don't. They eat like the rest of us, ultra-processed foods and a poor Western diet.
So in agreement with what Paul just mentioned, I would say that this is something that's happening also in Greece, that the traditional way of cooking, the way of the family having lunch, dinner, everything, this is lost. So in any case, I don't know what's your opinion, Paul. We have to re-educate the young generation, and of course, it's very, very important to remember that the diet has to also do with the economic status of the population, because the poorer you are, it's more prone to use junk food or to consume junk food. The guys which are very wealthy, normally they have like superb diets, right? So it's a multi-parametric story.
But it's a paradox, because the diets, like I'm thinking about the dieting on Crete. It was the famous Mediterranean diet that was initially studied by Ansel Keys. These were not the rich people. These were the normal people.
Exactly. But Roberta, you are right. They were not rich, but they thought there were not any alternatives, right? Yeah. So at those days, they had only, these things to be eaten. So now there are many, many alternatives, including hamburgers, French fries, and everything. So, at least the young people, including myself, sometimes they prefer, this type of food and not what we consider as modern diet. So, yeah.
But Paul, sorry, you go ahead, Brigitte, and then I will ask my question.
Yes, the time is ending, and I have to thank. We have to thank all the speakers for the clear answers for this debate on the Mediterranean diet. Should be organize another webinar about this. Thank you everyone for participating to this webinar. I hope this webinar has been informative and helpful. And I want to remind that recorded webinars are always available on the website. So share this information, disseminate it with your colleagues and students. And I'm looking forward to keep in touch for next events. So thank you all.
Thank you very much.
Bye-bye. Have a nice weekend.
Have
a nice weekend.
Bye-bye.
Thank you so much. Bye-bye.
Bye.